Okay, now let's talk about the process of gluconeogenesis. So gluconeogenesis is performed primarily in the liver, in the fasting state. Let's say that about 90% of gluconeogenesis in the body is done in the liver, and we have a small, minor amount that is done in the kidney, because the kidney needs to supply itself to ensure sufficient energy source. Now for gluconeogenesis, I like to start at the level of pyruvate. Pyruvate, of course, is the end product of glycolysis, and the sources of pyruvate are important to consider in feeding the gluconeogenic pathway, providing substrates for glucose synthesis. One source of pyruvate is lactate. The lactate is generated, of course, from red blood cells. Red blood cells, of course, have no mitochondria, so they must regenerate their NAD capacity to continue glycolysis by converting pyruvate into lactate. The other major source of lactate is skeletal muscle under anaerobic conditions. Anaerobic conditions means that we have to regenerate those NAD molecules for glycolysis to continue, and since we don't have oxygen present to continue the electron transport chain, we'll generate lactate. The lactate from red blood cells and skeletal muscle is then placed into the blood. And once the lactate enters the circulation, it arrives at the liver, and the liver specifically has transporters. These are anion transporters to take up small acid molecules like lactate. The lactate is then converted by lactate dehydrogenase in the liver, in the cytoplasm, and this is going to convert our NAD to NADH and result in pyruvate. The other source of pyruvate is from alanine. Alanine, of course, is an amino acid. The largest source of amino acid of alanine is from skeletal muscle. And recall that amino acids are not stored in a pool. So amino acids are typically in the body stored as protein because of protein synthesis, and there's no real pool. So here we are having skeletal muscle turn over protein, releasing alanine. Now the alanine is converted in the liver by an enzyme called ALT. ALT is alanine transaminase. We'll learn more about this enzyme and this reaction in the urea cycle. So alanine and lactate are the two primary sources, important sources for pyruvate. Now the pyruvate enters the mitochondria, as usual, we've seen before. And the pyruvate, once it's in the mitochondria, has two potential directions. The first direction is, as we've seen before, which is the liver in a fed state, and this is the conversion of pyruvate to acetyl-CoA by the enzyme pyruvate dehydrogenase. This, of course, enzyme is a oxidative decarboxylation reaction, generating some NAD from NAD plus NADH. The other avenue for acetyl-CoA, or excuse me, pyruvate, is to convert it into oxaloacetate. This is done by an enzyme called Pyruvatecarboxylase. Now, the question becomes, what decides the direction of pyruvate conversion? Does it go into acetyl-CoA or does it go down the oxaloacetate? The direction of this is largely going to be dependent upon the concentration of acetyl-CoA. The acetyl-CoA concentration in liver mitochondrial matrix is exceptionally high in the fasting state. Recall that the fasting state is designated by the hormone glucagon. Glucagon will stimulate adipocytes, our storage cell for fatty acids, to release fatty acids from triglyceride stores by lipase activation because of the glucagon signaling. These fatty acids now enter the bloodstream, enter the liver mitochondria through the carnitine transport system, and undergo beta oxidation to liberate acetyl-CoA. Now, in the fasting state in the liver, there is an enormous amount of fatty acids that are being able to be oxidized. This is because liver cells have a very high concentration of mitochondria. So the acetyl-CoA concentration in the fasting state in liver mitochondria is exceptionally high. What this is going to do is it's going to feed back, as we've seen before with the pyruvate dehydrogenase enzyme, and it's going to inhibit pyruvate dehydrogenase. So that is going to prevent the amount of pyruvate transforming into acetyl-CoA. Once we have a buildup of pyruvate, what do we do with it? Well, we have pyruvate carboxylase. In this case, under these conditions, high concentrations of acetyl-CoA will activate pyruvate carboxylase. That will turn pyruvate process into oxaloacetate. This is a biotin-containing enzyme because we have to add CO2. Recall that the pyruvate is a 3-carbon molecule, oxaloacetate is a 4-carbon. Now, once we have our oxaloacetate, we are going to convert this back to malate. So this is a reverse step of the TCA cycle, and this is done by the malate dehydrogenase enzyme. And recall, the reverse direction is actually favorable. When we looked at the delta G, standard free energy, we saw that this was a positive 7.1 in the normal TCA direction. That means that in this direction, the standard free energy is negative 7.1. So this is essentially a favorable reaction. What further drives the reaction from oxaloacetate to malate is that we are generating a large amount of oxaloacetate from the pyruvate carboxylase reaction. Because this enzyme is activated by high levels of acetyl-CoA, which are coming from beta oxidation of fatty acids, we are pushing pretty much all of the pyruvate in this direction. Now, once we form malate in this reaction, the malate is then going to exit the mitochondria. We'll talk a little bit more about this malate shuttle here later in a later video. But for now, let's talk about malate entering the cytoplasm. Malate, the first reaction, is going to be conversion of malate back to oxaloacetate. This is done by the cytosolic malate dehydrogenase enzyme, and this is going to be delivering reducing power to the cytoplasm. So we are reducing malate to oxaloacetate, and we are oxidizing NAD to NADH. This will become important NADH in one of the steps in reverse glycolysis we'll see in a second. Oxaloacetate then is converted to phosphoenol pyruvate. Recall, this is one of our enzymes in the glycolytic pathway, PEP. The enzyme that does this is PEP carboxykinase. phosphoenolpyruvate carboxykinase, abbreviated PEP-CK. Important about this enzyme is that this is a target of the anti-diabetic drug metformin. The rationale for this drug is to inhibit gluconeogenesis by blocking this step at PEP-CK, and by blocking endogenous glucose synthesis in the liver, we can try to assist a diabetic in maintaining glucose control by not adding to the problem. Now, once we arrive at PEP, we have to take a look at the forward reaction. Recall the forward reaction in glycolysis is accomplished by pyruvate kinase. This is our second ATP generating step in glycolysis and an irreversible reaction. So we have to prevent the PEP from going down this pathway in glycolysis, or we will have a futile loop. We will never be able to get these substrates into gluconeogenesis. So this is where glucagon comes in. Glucagon signals, of course, through G-alpha S, adenylate cyclase, cyclic AMP, and ultimately protein kinase A, our usual signal transduction for glucagon. pKa will phosphorylate pyruvate kinase, and in doing so, inactivate the enzyme, thus preventing the futile loop of going from PEP to pyruvate. Now, the series of reactions from PEP to 1,3-bisphosphoglycerate are reversible. And so as increasing substrates coming up the glycolytic pathway, that will be the thermodynamic energy to drive the reaction forward, since these are all easily reversible enzymes. The next step here is to convert 1,3-bisphosphoglycerate into glyceraldehyde 3-phosphate. This, of course, is done by glyceraldehyde 3-phosphate dehydrogenase enzyme, and this is where we will need the NAD that we've delivered from the mitochondrial matrix to the cytoplasm by our malate shuttle. In this direction, recall, is the opposite direction of glycolysis, and so to reverse this direction we are going to need NAD. So the NAD we delivered here in shuttling malate instead of oxaloacetate from the mitochondria, we have reduced this molecule and generated the NAD that we need here. We also have a source of this as we are delivering lactate into the liver from skeletal muscle, and once again here we are oxidizing lactate into pyruvate, generating NADH. Now the next reactions here up to fructose 1,6-bisphosphate are reversible. So all of these reactions from PEP all the way up to fructose 1,6-bisphosphate are reversible. We have a little detour here because we need to explain the reason why we need NADH in the cytoplasm. Recall that the concentration of NADH is very low in the fasting state in the liver. That's because glycolysis is not running. We have a block here, and we'll see the next block in a second. So the next reaction is to take our fructose 1,6-bisphosphate and convert this to fructose 6-phosphate. This, of course, is done by the enzyme PFK1, phosphofructokinase 1. Now, this is an irreversible reaction, so this direction is not going to occur. only the forward direction. Recall, because in this step we are harnessing the power of ATP to drive the reaction forward and phosphorylate the fructose on the number 1 carbon. So this is not a reversible reaction. So we need to go around this direction. So we have another enzyme that will bypass this, and this enzyme is called fructose 1,6-bisphosphatase. The job of this enzyme is to remove the phosphate on the number 1 carbon as inorganic phosphate and result in the product fructose 6-phosphate. Now the question is how is this particular site regulated? Well, we know that ATP is an inhibitor of PFK1. ATP levels are very high in the fasting state in the liver, primarily because we are undergoing enormous amounts of beta oxidation. So we are able to keep our ATP levels exceptionally high. This contributes to inactivating PFK1. We have another system that involves an enzyme called PFK2, phosphofructokinase 2. This is a unique enzyme that it is a double-headed enzyme, as I like to call it. One site on the enzyme has kinase activity. The other site has phosphatase activity. So from this, you can already begin to guess that this enzyme is going to be involved in phosphorylation, dephosphorylation reactions that will control another enzyme, that being PFK2. So the way this enzyme works is that PFK2 is responsible for taking fructose 6-phosphate and converting this into fructose 2,6-bisphosphate, not 1,6, 2,6. When it does this reaction, this fructose 2,6-bisphosphate is an activator of PFK1. So when there's plenty of substrate in glycolysis in the fed state, we have plenty of fructose 6-phosphate. We want glycolysis to be running forward. So we are going to take a little bit of this, make fructose 2,6-bisphosphate with PFK2 kinase activity, and then this is an activator to keep glycolysis flowing. Now, when we're doing gluconeogenesis in the liver, we need to inhibit PFK1. So we see that ATP participates in that function, but we also need to remove fructose 2,6-bisphosphate from this the chemical equation to remove the activator. And the way we're going to remove the activator is to turn on the phosphatase activity of PFK2. So the kinase activity puts the phosphate on, the phosphatase activity removes the phosphate. Now, how is this regulated? This again is regulated by glucagon. Glucagon will induce a phosphorylation event on PFK2. This phosphorylation is mediated, of course, as glucagon signals through pKa, and this phosphorylation event will inactivate the kinase activity and activate the phosphatase activity. So by doing that in a fasting state, we are now activating the phosphatase to decrease and remove the concentration of fructose 2,6-bisphosphate by removing that phosphate from the number 2 carbon, bringing it up to here. So the regulation of PFK1 is twofold. First, we have very high levels of ATP being maintained in the liver cell, in a fasting state because of the enormous beta oxidation of fatty acids coming from adipose due to glucagon signaling. The second level of control is PFK2. So in a fasting state, we have glucagon signaling, pKa activation, and that protein kinase A phosphorylates this enzyme, which inactivates the kinase and activates the phosphatase, so that now we deplete the amount of activator. Now once we get to fructose 6-phosphate, the isomerase, phosphoglucose isomerase will convert this to glucose 6-phosphate, and then we need to convert this to glucose. Now recall, we have a glycolytic enzyme here that is called glucokinase. Glucokinase is responsible for phosphorylating glucose, so we are going to be using ATP here. And since we're using ATP, this is a high energy reaction and it is irreversible. So we cannot reverse glucokinase activity. Rather, what we are going to do is bypass this step, and we are going to use the enzyme glucose 6-phosphatase. This enzyme is responsible for removing the phosphate from the number 6 carbon on glucose, forming glucose. Now, what is the preference between these two enzymes? How is this point of regulation controlled? It's controlled by substrate concentration. Recall that the affinity for glucokinase for glucose is very low. Under these conditions of fasting, glucose 6-phosphate concentration is going to be relatively high because we have lots of substrate coming up the reverse of glycolysis to generate glucose 6-phosphate. The glucose will exit through GLUT2, glucose transporter 2, from the liver and into the blood. Thus, because of this transport, this exiting, the glucose concentration under these conditions of fasting is very low. With a low substrate concentration, glucokinase is essentially inactive because it's a low affinity enzyme. There's not enough glucose for this enzyme to work. Conversely, we have a high concentration of glucose 6-phosphate, low concentration of glucose. This is a substrate for glucose 6-phosphatase, which will drive the reaction in the reverse to form glucose. Now flip this onto the fed state. Recall that the glucose 6-phosphate levels in the fed state are moving quickly down glycolysis. Glucose is high because we're in the fed state. We've just had our carbohydrate meal. And so now in the fed state, the concentration of glucose is high. Concentration of glucose 6-phosphate is low. So based on that balance of substrate product, now we can activate glucokinase, and glucose 6-phosphatase is not active because its substrate is disappearing into fructose 6-phosphate. Now one more point we have to take into consideration is back here in glycolysis we have the reaction of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, DHAP. This reaction, the reverse of this reaction, is the aldolase. DHAP at this point is very important because this is another source of gluconeogenesis. The source of this is glycerol 3-phosphate. The glycerol 3-phosphate by the glycerol 3-phosphate dehydrogenase enzyme is a redox reaction that will generate NAD that we'll need for the reverse glyceraldehyde 3-phosphate dehydrogenase reaction. But this reaction here will supply DHAP. So where does glycerol 3-phosphate come from? It comes from glycerol. The enzyme that does this reaction is called glycerol-kinase. This is, of course, an ATP utilization process. But even though we're in a fasting state in the liver, this is not a problem because all of the beta oxidation is supplying a high level of ATP. So this will not be inhibiting. Now the question is where does the glycerol come from? Well, let's go back to the very beginning. Glucagon-stimulating triglyceride hydrolysis by the lipases releasing fatty acid. Recall, the backbone is also being released, which is glycerol. This glycerol enters the blood from adipocytes, and then it is taken up by the liver, by hepatocytes. The hepatocytes now take the glycerol, use glycerol kinase to make glycerol 3-phosphate, and then oxidize the glycerol 3-phosphate through the redox reaction of the dehydrogenase to generate DHAP. The DHAP now enters the gluconeogenic pathway. So although we've seen the substrates of alanine and lactate are substrates for gluconeogenesis, they enter through pyruvate and have to go through this circuitous route. Glycerol enters at the level of DHAP, so it has a shorter route into glucose synthesis.